Hello guys, welcome back to a brand new video. Today we're going to be doing about enzymes and the role of ATP and NAD, leaving cert biology higher level. Before I start the video, make sure you check our channel for more videos like these, and make sure you like and subscribe. And yeah, comment down below for any questions. So let's begin. So, introduction to enzymes. Enzymes are biological catalysts that speed up chemical reactions in a living organism without being used up in a reaction. Now, the definition of enzyme is really important for leaving cert biology because it's a common question to be asked in a short question. So they could ask like, what is an enzyme? Enzymes are biological catalysts that speed up chemical reactions in living organisms without being used up in the reaction. Most enzymes are proteins. They are essential for metabolism and cellular functions. Each enzyme acts on a specific substrate due to its unique active site. Active site is when I'm gonna be explaining more about what active site is throughout this video. So really important what an active site is. I'm gonna be showing you the properties of enzymes. Enzymes are highly specific to their substrate, work best under specific temperature and pH conditions, not permanently changed or consumed in reactions. Enzymes can be reused, activity can be influenced by inhibitors or environmental changes. So you could reuse enzymes as many times as you want. So this is really important for an exam question. An exam question could be like, what the benefits of using an enzyme and the properties of using an enzyme. You could just say it can be reused many times and not permanently changed or consumed in a reaction and highly specific to the substrate. And how and for the other question, they could ask you, how can an enzyme work the best under temperature? So like they work the best under specific temperature and pH conditions. It depends on the type of the enzyme is. So like, and the activity can be influenced by the inhibitors or the environmental changes. So the where the enzyme is reacting the environment beside it, like if the light's too, if the heat in the room is like way over, the enzyme can react, or is it way too cool so the enzyme can't react. So you gotta put the temperature at the middle so the enzyme can react the best, and we can get the best results, and we can write it down in our scientific journal. Active site theory, really important for biology leaving cert. The active site theory explains how enzyme functions. Each enzyme has a specific region called the active site. The substrate fits in the active site like a key, a lock. So like when the enzyme reacts, it fits into the substrate, and then when the enzyme doesn't react, it releases, and it turns into two pieces. It's kind of like a key that fits the lock, and it gets out, and then but the enzymes are not being used up, and they remain unchanged. They become how they were before the enzymes were attached. So each enzyme has a specific region called the active site. The substrate fits into this active site like a key fits a lock and a key model. The enzyme substrate complex forms, leading to a chemical reaction. The enzyme then reacts the product and remains unchanged. Enzyme specificity. The specificity refers to the enzyme's ability to bind only with a particular substrate. Due to the unique shape and structure of the enzyme's active site, Slight changes to the active site, example due to the temperature or pH, can affect enzyme function. So the enzyme can only react the most due to, for an example, could be the temperature or pH, which could affect the enzyme's function and its unique shape and the structure. Factors affecting enzyme activity. Temperature, really important. Temperature and pH are like the most common things you could say about factors affecting enzyme activity. This could come up as a question for the Leaving Cert. Temperatures. Temperature, enzymes have an optimum temperature at which they work the best. pH enzymes. Each enzyme has an optimum pH range for activity. Substrate concentration. Increasing substrate can increase enzyme activity up to a certain point. Enzyme concentration. More enzyme molecules can lead to a faster reaction. So the more enzyme concentration reacts to faster reactions, and substrate concentration increases. Substrate can increase enzyme activity up to a certain point. So the highest the enzyme can react, and the pH can optimum the pH range for activity, and the temperature at which the enzyme reacts the best and works the best. Optimum activity and pH. Optimum activity refers to the conditions under which an enzyme works most efficient. For example, pepsine works the best at pH 2, acidic in the stomach. Amylase works the best at pH 7, neutral in the mouth. Amylase is formed as a chemical reaction to break down food and occurs in our mouth. Deviation from the optimum pH can reduce enzyme activity or denature the enzyme. Heat denaturation of enzymes. Denaturation is the irreversible loss of enzyme activity due to a structural change. High temperatures can break bonds in the enzyme structure. This changes the shape of the active site. The enzyme can no longer bind to its substrate. Denatured enzymes are permanently inactive. Denaturation is really important. This is how the irreversible loss of enzyme activity due to the structural change. This change also shapes the active site, and denatured enzymes are permanently inactive as well. Really important points. Investigation, heat denaturation. Objective: to investigate the effect of heat denaturation on enzyme activity. Materials: catalase. Example from celery. Hydrogen peroxide, boiling water, test tubes. Method: boil enzyme solution and test its ability to break down hydrogen peroxide. Observation: boiled enzyme shows no bubbling, no activity. Conclusion: heat denatures the enzyme and stops activity. This experiment is really important to learn off. ATP, adenosine triphosphate. I've explained this in more detail in the video of respiration, so make sure you go check that out. And this is a brief of the production and what ATP is and the nature, and NADP and the role of NADP and the role of and the importance of coenzymes. So ATP, adenosine triphosphate, really important. ATP is the energy currency of the cell. It stores ATP occurs in the mitochondria, which is also the powerhouse of a cell. It stores energy in its high energy phosphate bonds, ATP adenine plus ribose plus 3 phosphate groups. Energy is released when ATP is broken down into ADP plus P. Production of ATP. ATP is produced in mitochondria during cellular respiration. The equation ADP plus P plus energy is ATP. Energy comes from breakdown of glucose and other nutrients. ATP is used for muscle contraction, active site synthesis of macromolecules, etc., and many more. Nature and role of ATP. Immediate source of energy for a cellular process. Allows cells to carry out life functions efficiently. Is not stored in large amounts, constantly regenerated. Used wherever energy is needed rapidly. NAD and NAD+. NAD, nicotinamide adenine dinucleotide, and NADP plus coenzyme. Involved in redox reactions. Transfer of reactions and hydrogen ions. NAD, which is used in respiration. I posted a video there just before this, a video about respiration. Make sure you check it out. Really important topic for leaving cert biology. NADP plus is used in photosynthesis. Role of NAD in respiration. NAD accepts electrons and hydrogen ions during glycolysis and Krebs cycle. NAD transports these electrons to the electron transport chain. This process helps generate ATP. NAD is recycled for use again. The role of NADP plus in photosynthesis. NADP plus is reduced to NADPH during the light-dependent reactions. NADPH carries high energy electrons and H plus ions to the Calvin cycle. It helps convert carbon dioxide into glucose. NADP plus is then regenerated and reused. Importance of coenzymes. Coenzymes are like NAD and NADP plus are essential for enzyme functions. They assist in electron and hydrogen transfer. Without them, many enzymatic reactions would not occur. They help maintain energy flow and redox balance in equations. Summary of key concepts. Enzymes are specific proteins-based catalysts. The active site theory explains how enzyme works. Temperature and pH affect enzyme activity. Heat can denature enzymes irreversibly. ATP is the universal energy carrier. NAD and NADP plus play critical roles in metabolism. Importance to living organisms. Enzymes make life-sustaining reactions occur quickly enough. ATP powers essential cell processes. NAD and NADP plus are essential to respiration and photosynthesis. Understanding these molecules help in medicine, industry, and research. Thank you guys for watching. Make sure you comment down below for questions, and please make sure you don't forget to like and subscribe. And I'll see you guys next time.